whenever you're ready, just please go ahead, and thanks again for joining us.
Great, thank you, Jenny, and good morning, everyone, or good noon if you're in the East. Happy to spend a little time with you to share the browser a little further. Before I get started, though, I don't know if there's enough people on the call yet to ask the question, but I'm interested to know how common it is for you all to look at variants that are not in genes. Do you believe there's enough information to help you with variants and enhancers and promoters and so forth? I'm going to pause for a few seconds and give you a chance to reply.
So this is Jenny. I can take a stab at this, and anyone else, please feel free to jump in as well. So I would say in the work that we're doing, we are typically looking at variants in coding regions or splice site junctions. So really looking at variants for which there is evidence that a variant can be associated with a disease. But I mean, certainly if a paper had reported an assertion that there might be a promoter variant or something, in an intron, something like that, that's associated with a disease, we would look at that. I mean, most often, as I say, it's coding region, splice site junction, but if an author has made that assertion, we would definitely look at that too.
Great, thank you.
For other gene areas.
And my other question is, do any of you all have any questions before we get started? Anything maybe that came up last time that you wanted to follow up on or get some clarification on, or anything specific you'd like me to address before I plunge in?
No.
Okay, I'll take that as a no for the moment. Do please feel free to interject at any point if you have a question about something. I know when I'm listening to a talk, I often have a question and then I can't quite formulate it when the time comes to ask questions. So if we can do it in real time, that's fine with me. So let's get started. A nod to the funders. The NHGRI is our primary funder, and a few other places have some money that comes into the project. These are the people who do the work. The engineers are a creative bunch of programmers. The QA people are really good at breaking the browser and keeping the engineers honest. And they're kind of the watchdogs of the user interface. They help make things a little bit more usable by being more like a user maybe than an engineer is, more like an end user. And Jorge, Eric, and Haifong keep all the hardware running. Here we are on the central coast of California. And here we are in the trees in the central coast of California. And there I am in the back, since we're not on a place where we can see each other. Okay, somebody's mic is on. Okay.
So please, if you could please mute yourself when you join the call, just so we don't have background noise. Thanks so much.
Of course, if they haven't joined the call yet, they're not hearing you. Okay, so we have a training channel we have about 16 videos, we have 16 videos now that show you some of the things that we think are most easily missed by people using the browser. A number of different topics. We just released one a couple of days ago. And we have on-site workshops where we can come and teach your whole university or your whole department in groups for a full day or 2 days. You can find our schedule there and or sign up for trainings. One of the things we really like about those is we get to watch people work. We get to see in a little bit of one-on-one time how we can improve the browser by interacting directly with people. So the browser is a display engine. We have tons of data sets. We have a coordinate space that allows us to hook up anything that can be mapped to a coordinate, such as a histone modification site, a mRNA that's been discovered in one tissue or another. And we have a consistent interface across more than 100 animals. But of course, in the context of human curation, it's the human genomes that are the most important, and most of our users are on human. The human genome we have a half a dozen different releases of the human genome from HT16 up through HT38. so that you can work at any, if you have a project on HT18 and you haven't yet moved to HT19, when we do, we don't throw away the older version, so you can continue to keep the coordinate space and so forth. Or if you read a paper and the paper refers to coordinates on an earlier version, you can go to the browser and jump from that earlier version to the most recent version and back and forth. So here's a few of the objectives for today. I don't know if we'll reach all of them, but I want to show you multi-region mode, especially exon only mode, which is the reason why I ask you if it's common to look for variants not in genes, because exon only mode gives you the opportunity to throw away the introns and the intergenic regions and just look at the parts of the genome that are most important for a whole exome project or an RNA-seq type project. You can also load a list of regions such as a pathway or something like that, where you can just look at a handful of genes, whether or not they're contiguous in the genome. I'm going to talk about several different data sets that are likely to be useful to you in your work. A number of different ways to navigate in the browser, different ways of inputting data. Some of this will overlap with things I talked about last time, but for the benefit of people who weren't there and maybe to help clarify some things. I hope it's not too repetitious, but a little bit of overlap. I want to talk about the SNP tracks that we have, the difference between our all SNPs track and our common SNPs, how to save sessions, and how to recall sessions and use them to facilitate your work and getting back to your train of thought. I don't think I'm going to talk about beacons. We'll be talking about Gtex, and if I get to it, we have some spreadsheet templates you can download that make it easy to have a link to the browser in a spreadsheet that lets you jump directly to a region of interest or a gene or something like that. Oops. So here are some links that are relevant for today. The bottom link is an appeal. If you have any programmers among your acquaintances, have them give us a shout. We're looking for a few people to help us keep the browser going. Okay, one of your number last week, one of the few questions we got last time was a question of how long have we had HGVS input as a way of navigating in the browser, and it's been open, it's been available for more than a year. And I wanted to let anybody know who was not here last time that that's a really good way to get around in the browser. And if you missed the announcement on it, you might not know that it's available. The bottom link down here takes you to our help page, which outlines the number of different types of HGVS input you can use. We don't cover the entire gamut of HGVS, which could get quite complicated, and some of which gets beyond the ability to display it in the browser. But many of the basic types are available now, and you can type in any of these formats, either cDNA type or protein type, or even illegitimate HGVS, which is just an amino acid number, can take you to the correct amino acid in the gene. So now I'm going to switch over to a live demo on the, right there on the Genome browser. Here is the home page. If you're following along, reset all user settings is a good way for us to start at the same location. And wait for a second while it reloads here. Why is it doing that? Things were very slow for me when I logged on. I think maybe the wireless in this part of the building isn't so good today. That's weird. That happened last time too, that we had a...
It's probably because you're trying to do a live demo.
it could, yeah. It could be that, but in general, it's very reliable. I mean, I had to find a quiet place in the building, so I let myself in my boss's office in the corner of the building. So maybe I'm farther away from the Wi-Fi than I usually am in my own desk. But okay, I just have to wait for a second. It's just downright ugly. Okay, well, while we wait for it to load, if you are playing along yourself. Okay, dare I try to reload it? God.
This probably doesn't help, but I know our DNS server is out right now. I'm in Spokane watching this, so a little bit on the West Coast, but not on the coast. But yeah.
Well, maybe it's the WebEx. Maybe it's slowing things down somehow, trying to broadcast at the same time as. There we go. Okay. 30 seconds can be an awfully long time when you're waiting for something like that in front of people. Okay, so here's the browser, the main page. This is actually the gateway page. The main page is back here, and there's nothing on there of too much interest if you're a regular user of the browser, except for the announcements, which is the way you would have found about HGVS if you had been subscribed. We have up to 3 or 4 a month emails if you subscribe. Other than that, you can check this page for the latest information. So clicking into the browser to go to our gateway where we choose the organism. We did reset all user settings already, which means HT38 is the default. So I'm going to switch to HT19 where the best annotation is and where most people I've talked to in recent years are still working. But you can see that if you get information from one of the earlier genome assemblies, you can go into there and jump from one assembly to another, and I can show you how to do that in just a minute. For now, though, I'm simply going to jump using the Go button into the main browser page, which turns on a number of data tracks at the default location, and eventually it turns on some buttons down below the graphic. I'll bet it's the WebEx. This just never happens. I can work all day and hit the browser dozens of times a minute, and it doesn't do this. All right, well, it is what it is. My first step is I want to hide all. There we go. Oh, there we go. Look at that. I can make the buttons appear. Magic. Okay, so I'm going to hide all and simply turn off all of the browser tracks and start with a clean slate, and then I'll turn on the genes track here to pack at the default location. So I want to show you a few, whoops, and then hit refresh. And then I want to show you a few ways to navigate in the browser. For example, you can type in an RS number, RS111033597, and hit go, and it will give you an option to choose which track you want to turn on. And usually you would choose the most recent version of the different releases of dbSNP. We keep the old ones around for the same reason we keep old genomes around. People get in the middle of a project and they don't want to see their data disappear. So we leave the old ones around, at least half a dozen anyway. And so this is the all SNPs track essentially. It has all of the variants, and the one that we clicked on is highlighted. And you can see here that it's in the middle of a gene, and it's red because the amino acid is changed. And for any one of the SNPs in the all SNPs track, if you click into an item, you get to a page that gives you a lot of details about it. You can see that the reference allele is the same as the called the ancestral allele. It's the same as the alleles in 3 closely related primates. But the observed allele is a C and a T, so there are humans that have the C. Other information, because it was red, that tells us that we have a change of an amino acid, and we have a missense from an isoleucine to a threonine by changing the middle nucleotide here. And we typically give it to you in a variety of different annotations, because the different isoforms in the UCSC Genes tract give the opportunity to see whether it's a missense variant in all of the isoforms. You can see in this case, it's a missense in 4 of the isoforms, but it's an intron in another gene that spans the region, which looks like it's a read-through transcript where you actually have 2 adjacent genes being transcribed together by some kind of a read-through. If I go back now to the browser using the Genome browser link, you can see at this resolution, you can see the amino acids in the the reading frame. You can zoom into a particular region by putting your mouse over the top of the track and zoom in. And when you zoom in close enough, you can see the amino acid numbers, and each one of the isoforms has a different number if there's a splice variant event that's happened upstream. So it gets out of...
Sorry, I have a question. You're showing the UCSC genes. Do you have the numbers also on the Refseq genes, or is that only for the UCSC genes?
We do. Let's turn on the Refseq Genes track to pack. And here are a lot of different, wow, I had no idea there were so many Refseq genes in many of these genes. The configuration button here on the left gives the opportunity to see what kinds of choices there are. So you can turn on by codons, show codon numbering. Okay, it's on by default if you're showing codons. So if I hit submit now, we should pick up the numbers. So here we go. Here are the
all right. Thank you.
Here are the amino acid numbers. Right. The HGVS nomenclature that I showed on the earlier slide. Let's go back to that slide and find it. So if you have an actual Refseq identifier, such as this one, that's a nucleotide identifier. You can use the HGVS as a navigation aid and simply put that in. And in that case, it will use the exact NM number for the transcript that you used and highlight that nucleotide. And it will show you this is that was a G to T, you may have noticed. And you'll see here that the genomic coordinate is a C, and that's because we're transcribed on the opposite strand. So it's coding right to left. So if you want to see it in the same orientation, you can flip the orientation by hitting the little button. And you can see now it's annotated as a G. So the software is smart enough to recognize that this transcript is on the opposite strand and look for the G in the right location. And it's highlighted in this vague kind of beige color here. I'm not sure how well that's coming through on the WebEx. You can, however, take any region and highlight it by highlighting the region like that, and you can give it a really obnoxious color if you like, and it'll change the background to any color you choose, which can be handy if you turn on a bunch of data tracks and you scroll down to the bottom of the page. It's a great way to keep track of where you are. For example, if you turned on a gene expression track such as encode RNA-seq, let's just turn it on to show. This is some expression data from the ENCODE Phase 2 project where UCSC was the data curation center. And if you just turn it on to show, a number of the tracks will turn on by default. There are something like 50 data tracks in here. And so here you are, scroll down the page, and you can see that there's not a lot of data in this region, but your highlighted region is very easy to keep track of. If you right-click on the highlight, you have the opportunity to remove the highlight. Okay, I'm going to actually turn off these tracks as well by right-clicking on the data controls on the left side here. And this particular region is not too interesting with this data set, but I do want to use a subset of these data in a little bit when I show you the exon only mode. As long as we have the all SNPs track turned on here, let's zoom out. Let's see, we have 11 base pairs in the window here. So let's zoom out by 100x and get out to 1100 bases. You can see that the codons are easier to see. Well, not easier to see, but at this resolution, we can't tell you the amino acid names, but we can still tell you the size of the exon, so you don't lose track of your scale. You can see how many amino acids there are. Switching the all SNPs track to dense, it throws away the names, and it just gives you the tick marks and shows you the coverage of the track. Now, one thing we did, many versions of dbSNP ago, so here we are on all SNPs 150. Back when HG18 was the most recent version and we were displaying SNP 128, must be 8 or 10 years ago now, we learned that people were using the SNP track as a filter with their sequencing experiments because the word polymorphism implies that it's a common variant in the population. And it's profoundly not true that the dbSNP is just those kinds of polymorphisms. In fact, 90% of the data in dbSNP are more rare than the 1% threshold. And so the all SNPs track, if you put the mouse over here, it simply says simple nucleotide polymorphisms. It's a little bit of a kludge because the track includes short insertions and deletions. So single nucleotide polymorphisms is not actually true. And dbSNP used to call it that. Now I think they've changed the name entirely, and they avoid the SNP acronym. The common SNPs track is a filtered track that we began putting into our database. At that time, our engineer Angie Hinrichs, when we learned about the fact that people were removing all of the SNPs from dbSNP, we wanted to give people a chance to see that maybe that's not the right course of action, that the common SNPs track is a 1% filter. And so if I refresh the track now on the 150, you can see how common the common SNPs are, not very common actually, compared to the full data set. And if you actually look at the numbers in the tables, it does come out to be about 10% of the total. So these are the SNPs that are 1% in the population, and they're also have to be in the database, but by being contributed at least 10 different chromosomes, at least 5 different homozygotes had to be sequenced, because some of them came out to be 50.000%. And it turns out one person was in there, maybe James Watson was in there once as a heterozygote and nobody else. And so dbSNP calls it 50% in their automated pipeline, which is decidedly not true. It's definitely not the minor allele frequency in the population. As long as we're talking about common SNPs and other kinds of variants, we have a number of different, we have another subset which are called flagged SNPs, and these are SNPs that have been flagged by dbSNP as being clinically relevant. Not necessarily that they're known to be causative, but they're in a gene that is known to be clinically relevant, that it's possible to have a deleterious mutation in there. Another data set that we have 1,000 genomes data. Let's look for phase 3 variants. I'll turn that on to dense as well. We have the EXAC project, and we're working on the NOMAD, which is kind of EXAC on steroids, more genomes and a lot more intergenic regions. And then finally, the DGV structural variants. These are the large copy number variants. This whole region down here has a lot of different database information. For those of you who are doing microarray data or looking at it, you can actually see the footprint of individual SNP arrays by turning on this track. These are commercial arrays that are available, and you can see the where the probes are in those arrays. That's handy if you're looking at a large copy number variant because it helps you identify where the endpoints are. If your variant stops at a certain spot, you don't always know just how much fuzzy area you have between the last probe and the next probe. So I've turned on a number of these other tracks now. So here's 1,000 genomes track. Let's turn on the flagged SNPs track. We'll put it to dense as well. And you can see that they're concentrated in the exons where you would expect them to be because they're flagged as clinically relevant. Here's DGV. There's one variant that runs through this region. These are supposed to be benign structural variants. It's not 100% clear to me that that's really true. Some of them may not be, may have been miscalled. There's a lot of old data in DGV. And then here are the EXAC variants. You can switch that down to dense and collapse it into the track that looks like the other ones, so you can see the density of coverage there. So we have a number of different data sets, and so let's say you get the browser configured just the way you like it, the data sets you're interested in. Let's say, for example, you don't like DGV because it's too big and too messy, so you can you can hide that. And so here you are, you have a configuration that you really like, and you want to keep track of that configuration. You want to be able to get back to the browser after you've browsed around with just this kind of view in mind. We have the saved sessions functionality here. I wonder if it works if you can't read it. So the first one, it did. Okay. So I'm going to sign out. This is from a workshop I did on campus here yesterday. And so we can log in with ClinGen 2018, password Genome, and log in. So any one of you could log in using this login, bearing in mind that it's a shared login, that anybody else who has it can see the login. You can also create your own if you like, and we send you an email and you have to click the link. And so, you know, the authentication process to keep the robots from making too many data tracks. And so I can save this. I can save this as, let's call it small benign SNPs, and create a session. So by submitting that, anybody who's interested can click this link and go there. Or if I use my right mouse button here to copy the link location, I can open up a new browser. Here's just another generic browser. Yours probably opens to Google. Mine opens to the Genome browser. You can put that link in, and you can email it to somebody else. And so here's a look at the browser. It looks like it's running off the page. That's weird. The resize button makes it fit the screen. That's kind of a bug. It's supposed to record the exact width of the screen as well when it saves the session. So now any one of you who's interested in could navigate to a screen that looks exactly like this simply by loading a session. And you don't have to be logged in to do that, because if you go to this page without being logged in, the restore settings option is still there. So you can type that information in, small benign SNPs, and you submit that, and it will take you to that location, whether you are logged in or not. And so you can share links with other people. You can keep track of, see it resizes each time. You can keep track of a particular view that you might be interested in getting back to. If you're interested in large regions and you're zoomed way out and you have other data sets turned on, you can save a session like that. And then, of course, you can navigate to other places. So let's navigate to a gene MYH7 to pick one not quite at random because of a conversation I had with Jenny at the beginning of this this call. And so here we are in the MYH7 region with all of those data sets turned on. So we're at 22 kilobases, so there are a lot of variants in the region. Here are the exact calling regions, places where there are exact data, and as you can see, they line up with the exons as you would expect them to. I want to load a session right now that shows you a little bit about our expression data and at the same time our exon-only data. So, or exon-only mode. So I'm going to go to my sessions, and you don't have to be logged in to do this, but if you restore settings using username example and HG19 underscore exons expression 3. So this is a session that's being loaded from one of my alter egos, which is example when I'm doing a workshop. You can see it was created on a narrower screen for a workshop on a, with a VGA projector. And so, but if you want to make it wider, you can resize it if it's the width of the screen then. And what we have here is we have 16 data sets in addition to the gene set. Each color represents a cell line, and each pair of tracks in the 4 of the same color represent one strand of the genome where the RNA has aligned. So you have replicates here on the minus strand. Minus is above plus because M comes before P alphabetically. And so you have a replicate here, You have nothing on the other strand, as you'd expect, because of the transcription being from one strand typically and not the other. And so this ATP11C gene is expressed in all 4 of these different cell lines. There are a bunch of other cell lines available, and if you click into the configuration button on the left side here, you have the option to turn on a lot of other cell lines, which I won't do at the moment. But it does show you that this particular gene is expressed in these 4 cell lines. Over here, the FGF13, fibroblast growth factor variant, this particular cell line has no expression in that, on either strand, and these other 3 cell lines do have expression from this gene. So here you have genes that are expressed in different tissues. Gives you the option to see if you're looking at a gene in a phenotype, is the gene expressed in a tissue that makes sense with the phenotype that we're looking at. Down here also, if I scroll down, you can see this is the RNA-seq. This is the track I turned on earlier in that other region that didn't have much signal of interest. We also have the Gtex track. I'll turn this on to full because it shows up as a graph. The Gtex track is another type of data that we have showing expression, and it's showing a graph here that's not too visible because of so many genes on the screen. It's a little bit hard to see, but we can click into this one, for example, and we can see that the FGF13 is expressed in the brain in a variety of different tissues here, not very well expressed most other places. If we had been zoomed in, we'd have a taller graphic that's a little bit easier to see, but I'm not going to do that right now because there's something else I want to do at the on that screen. So I'm going to go back to the browser, and I'm going to go up here to View Multi-Region, and we get a menu for a number of different ways to slice up the screen. I'm going to start here with Show Exons, and I'm going to leave the padding up for 6 bases, which will typically pick up a splice site on either side. If you're interested in some of the other splice binding sites, you might want to make this a little bit bigger because there are some conserved regions near splice acceptor sites that might be useful. That's something that we plan to include in the browser sometime in the next year or so, is better splice predictions for some of the, like the lariat formation site and so forth, things that if they're mutated might mess up splicing. So I'm just going to click the Show Exons and Submit. Whoops, before I do that, I want to draw your attention to the size of the region. It's 2.1 megabases of DNA here. And now I'm going to hit Submit, and the software is going to slice all 16 of those tracks into little slices and then reassemble all the slices into one screen view here so that only the exons are showing and the introns are gone. You'll notice that we're down to 27,000 bases here, 27 KB. It's between 2 and 3% of the 2.1 megabases that we had earlier, but if you're just interested in exons, it's got all of the interesting stuff showing. You can see up here where the little red tick marks are how much of the genome it covers, and that it does it discontinuously. If I scroll down the page, here's a better look at the Gtex tract now that we're zoomed in. If I put my mouse over it, you can see what the individual tissues are. This graph looks a little different than the other graph because one of them is in log scale and the other one is not. I'm going to use the right mouse button to hide this track now and then just look at the gene expression tracks. Here's the ATP11C track. Shows you a little bit of difference between the replicates in this one that wasn't obvious when we were looking at the key view, showing the exons because the exons are such a small portion of the display when the introns are visible as well. The little pink lines are showing your splits. This is a 12-base gap between adjacent exons right here. Over here on FGF13, you see something really interesting. This GM12878 cell line is not expressed. I'm sorry, it's not expressing this gene, but this stem cell line here is expressing off of this isoform that has this extra large exon on the 5' end of the gene and a really large intron. I'm sorry, a really large untranslated region, which overlaps the intron of these other isoforms. So this version of the gene, or I'm sorry, this cell line is expressing this version of the gene, and these other two cell lines are expressing off of this other set of exons upstream over here. So you have a nice example here of differential expression of different splice isoforms that's made really obvious when you're looking at exon-only mode, but not so obvious when you're looking at a whole gene or a group of genes in a region. I'm going to type a question mark right now to show you these keyboard shortcuts that we have. That pops up the menu that shows you some of the shortcuts. If you're doing similar things a lot of times and you're not crazy about pulling down menus or looking for buttons on the screen, you can do a number of different things, zooming in and out and so forth. The one that I use the most, or really only one I use, is default view when I'm getting out of exon-only mode. If you just type DV, it throws away the screen splicing and gives you gives you back to default view. It shows you where you were before you put it on to exon-only mode. I want to open up a new window now on my browser and go back to the PowerPoint and scroll up to here and pick this URL off the page here, bit.ly /ClinGen 2018, because I want to pick up a group of genes off that page. Whoops, let me use a different one then. I don't have it on that page. Let's put UCSC 2018, and I used it yesterday in that workshop. So here's a small gene list that a number of different genes that may or may not be associated with each other. I'm going to go back to the browser now. Go back to View Multi-Region. Whoops, no, I'm not. Never mind. I want the coordinates, not the gene names. So let's go back, not the gene list, but the gene coordinates. So I'll open the coordinates. These are the coordinates for those genes. Yesterday in my workshop, I showed people how to grab those coordinates out of the table browser. But for today, we're just going to use the coordinates directly. I'm going to paste the coordinates in, and you'll notice that they represent a number of different chromosomes. They're not contiguous. They're not on the same chromosomes at all in the genome. I'm going to use the highlighting to highlight alternate genes, because we have 5 genes in the list. So 2 of them will be one color. We'll ignore the warning. And we have a kilobase right in the middle here. This is one of the genes that was on the list. So you may be interested in looking at a pathway. I'm going to zoom out. And so you may put the genes of a pathway in such a list. And you can look up the coordinates. Actually, the process for doing that is available on one of our videos, how you would look up the gene coordinates, starting with a list of genes. And so here we have the 5 genes. This one's from chromosome 4. This one's from chromosome 8. This one's from chromosome 10. They're in the same order as the coordinate regions were that I pasted in there. And so you can see here that all of the data tracks are sliced up. We have a view of the screen now that represents it's a virtual chromosome that's 285 kilobases, and it has the gene positions from the input regions. And so any of the genes that you have typed in there that you're interested in are visible on the screen. Is that a nascent question, or somebody playing with the mute button? Okay, so maybe it's not a question. I'm going to use the right mouse button here and close this track set turn off the gene expression data. And since I mentioned pathways, I want to show you a new pathway tool that we have. If I can find it, that's okay. So let's use the track search functionality here. So track search lets you use a keyword and find data tracks that may be relevant to something you're thinking about. You may be interested in histone modification. You could put you know, a methylation in there and get hundreds of tracks. You can turn on pathways. And so here's protein interaction data tracks, which is the one I was looking for. We'll turn it on to pack and turn it on. And so here is a data track that was released within the last year by our engineer, Max Heusler, generated by curating the literature using automated processes looking for keywords such as gene A modifies gene B, or controls gene B, or inhibits, something like that. And it generates for each of these genes a list of genes that are associated with it in a pathway. And if I click on that, it brings you to a matrix that shows you the nearest neighbors that are associated with that gene in terms of how that gene might be functionally interacting with it. So this is a very powerful interface. It's very very complicated and very very useful once you get the hang of it. And I have to admit that I don't actually have the hang of it yet. I haven't spent a lot of time with it. But there's an expensive, time expensive, but extensive help page here for interpreting the browser. So if you put your mouse over any one of these links, it tells you that there's text mining. There's 41 abstracts that associate MAPK1 with IGF1. In this case, the relationship between FGF and MAPK3. And this page is actually interactive itself as well. You can click on any one of these genes and it becomes the central gene in the region. So you can see genes that are purported to have relationships with each other. If we go back to the genome browser by clicking that link up there, here we are back on our page that has multiple genes on it. And I'm going to hit default view here, type in DV, and it goes to the page with the leftmost gene of that group visible. So the FGF2 gene is turned on. So for any of genes on the browser, you can turn on other data tracks that we have. For example, on the phenotype and literature track, you can see whether OMIM has any individual single base alleles that have been annotated. You can see if there's been a gene reviews article written for the gene and turn that on. And in case of the FGF2 gene, that appears not to be true for either one of them. You can navigate by typing just the name of a gene, NF1, and navigate over to that gene, and you can see here that there is a gene reviews article. So for any gene for which there's an article, so there are three articles here that are related to NF1. The neurofibromatosis 1 article itself is available directly by clicking on that link. Any of our details pages links typically open up a fresh window. So if I just jump back to the previous window, I haven't lost my place in the browser. As in any of our tracks, there's a track description down here that describes where the data came from, a reference to the original data set or data contributors and so forth. So if you're writing a paper and you've used browser information, it's a quick way to get back and get references. Or if you want to actually question the veracity of the data. You can go back and read the paper and decide for yourself just how well documented the data are, and do you believe the the suppositions that are proposed for it. The OMIM allelic variants, I like these a lot because the individual variants are available there. If you click into this one, it's a TRP2 terminator at amino acid 1538. And if you click into the OMIM allelic variant link directly, it brings you to the OMIM tract, and it gives you the reference where that individual variant was identified. Now, we don't have all of the OMIM variants because it's not easy sometimes to map the variants based on the information given in OMIM. But if OMIM has associated it with an RS number in dbSNP, then we can use the dbSNP coordinates to map the variants. And so we have anything with a dbSNP variant in it is available on the browser, and you can get directly to the article. 20 children in this paper on the NF1 gene. As before, you jump back to the browser by either closing the window or navigating away from it. So if I click now back to the genome browser using forward navigation, you can once again, you can save a view like this if you're interested in a large region and you want to know which genes are in the gene reviews, for example. You can type in something like Prader-Willi and hit go, and you'll find a large number of options. Because Prader-Willi is not actually the name of a single gene, the drop-down menu did not prompt you with a gene name, but the interface takes you to a list of genes for which the phrase Prader-Willi appears somewhere in the short description or even the longer description of the gene in the Refseq document. For example, here it's not in the short Refseq, so it must be in the larger, the longer Refseq. We can click on one of these genes, PWRN1, for example, navigate directly to the browser. The one I should go back here. The ones in bold are the canonical gene, the one that's got the most evidence for it and has the highest rating in Refseq as having been validated. The other ones, there are other variants of the gene sometimes, and they would be in, like this one, for example, is not highlighted, it's not in bold. So if I click on that gene, the one that was in bold is highlighted, and you can see that we're here on chromosome 15. I can turn on the chromosome band track here to pack, and actually I didn't have to do that because up here in the ideogram it shows me that I'm in Q11.2, so I'll just leave that turned off. You can type in 15Q11.2 if you're interested in that specific cytoband, and then the browser will navigate over there and show you all of the genes in that region. It shows you in the red ideogram up here where you are on the chromosome. Now, at this resolution, when you're zoomed way out to 5 megabases, you've got a lot of isoforms here that are maybe not interesting to you because, if you have a deletion or duplication, all of the isoforms are deleted, so you're not as interested in that. The configuration track for the UCSC Genes track, or the configuration page, if you click on the little button on the left side there, gives you the opportunity to turn off the splice variants and look at just one gene per locus, or one gene variant per locus. You can also turn off non-coding genes if you're mostly interested in protein coding, and hit submit. And the result is a page that's a lot cleaner. There's a lot fewer gene isoforms in it. if you're interested in taking such an image and saving the image, I highly recommend just making a session out of it, because then you can navigate to other parts of the genome and still have the same data tracks turned on. You may have interest in these same data tracks with the same configurations for other genes. You can jump around in the genome. If you want to make a static slide out of it, though, View PDF gives you the opportunity to make a PDF of the current browser graphic. And this PDF is actually, it's a vector graphic image, and so it can zoom to a lot of different scales, including the possibility of saving it to your own local machine. Then you can manipulate it in Photoshop and save it as a 300 dots per inch image for a publication. On the browser itself or on a display for a talk, it's fine to have screen resolution and do a screen grab, but if you try to reproduce it on a poster and you want it to be easily visible on the poster, you have to shrink the image to get it crisp, unless you're in PDF. So the ability to export as a PDF is a handy way of getting a high quality image that you can save at really high resolution and print it blown up on a poster if you want to have a big image on the resulting print version. So let's see, something else popped into my head just a second ago and now it's gone. Okay, just had a brain erasure, but it's a good time for it. I see we have 7 minutes left. It's a good time for me to pause and give you an opportunity to ask some questions before we log off.
Any questions for Bob? So Bob, I wasn't aware of the protein interaction resource on UCSC. That's really great. So you mentioned that this was done by literature mining, so looking for words like interaction or that type of thing. Do you know, I mean, does this include predicted interactions as well as known physical or biochemical interactions? Because this is data that's really, we use this a lot in gene curation. If a gene product interacts with another gene product that's already associated with that disease, we can give that some weight. So I was just curious to know a little bit more about that.
I can't answer that question myself. I suspect it might include predicted interactions. It does give you the option to go find the original literature and interpret for yourself whether it is or not. I will check with Max and or read the help docs myself. I guess I should do that first, huh?
All right.
So I was just playing around with it, and I saw that, so I had asked a question about, like, I want to be able to use only the abstracts, but you can sort by those that have abstracts. So for instance, with VHL, there were probably like 100 genes that were associated, and all of them said like their source was one database interaction, which isn't really useful for us. But if you sort by abstract, then the top 2 genes had like 2 and 3 abstracts associated with them. So that would be very helpful. It doesn't help us in terms of, which assay or, if you're, specifically trying to curate for functional information only, you can't really get that from there, but it limits it to 5 abstracts you need to look at for that whole list. So that's very helpful.
Yeah, it's fantastic. It's a really nice resource. Yeah.
Wonderful. That's great to hear. We have other pathway information also. If you click into a gene and look at its details page, down on the page here is pathways. We have the Kyoto Encyclopedia of Genes and Genomes. This is a little bit old. We don't have access to their behind their paywall because they lost their funding to make it free. But in this case, you can look at jump through to an individual gene and see some of the pathways as they've annotated them at Kyoto. And depending on the gene, we have 2 or 3 other databases that also give you pathways. So here's the VGF-A, VGFR2 pathway for this particular gene. Just picked one at random here. And the reactome pathway also gives you some pathway information that you can get to. I like the graphical pathway of the Kyoto quite a bit. And in fact, if you go from an ATPase gene and go to the graphical pathway, it really warms my inner biochemist to find their pathway shows the entire mitochondrial membrane with all of the cytochrome electron transport complexes in full glorious color, with the rotor and the proton pump and so forth. So it's a really really beautiful pathway. So we have a number of different pathway options available, but I'm very happy to hear that at least one of you found the Max's pathway track really useful. I freely admit that I haven't spent enough time with it, but now that I have an endorsement from y'all, I'm going to spend more time with it and maybe feature it more prominently in the future.
Great. All right. Well, I
think now that we know about it too, I mean, we can use it as well. I think there's so many options to get data through the UCSC Genome browser, and what I find is just knowing the path. It's like going through a large file tree or something. It's like you have to know the path to get to that track or something. So it's great to know that the PFAM track in UCSC Gene is another one too that I think for biocurators in particular, having that PFAM track up helps you to know what protein domain you're in. You have to have I think you have to have UCSC Genes on, or it's the PFAM is mapped to UCSC Genes, not to Refseq Genes. I think that's the issue with that.
Okay, so the PFAM track, this is excellent feedback. I love hearing from people who are on the front lines using the browser. So here's PFAM domains, but this is just gene by gene. Let's go back to the browser and let's use track search to type in PFAM. Here's PFAM in UCSC Genes. Okay, so let's turn that on. And so once you have found a track you like, you can save it in a session, so you don't have to find it anymore. That's one of the beauties of the saving a session functionality. So you can turn on just the track. So here it is in the genes and gene prediction track, and so you can find just the tracks that you like and turn them on, and then you can get back to them. I know what it was. It was the Excel spreadsheet. We didn't get to that. Maybe version 3 in a few months. If you're not tired of hearing my voice, we could do another thing. I could spend more time with Max's track and spend a little bit of time with